let's come to the next one, that is cytoplasmic genetic male sterility. This is cytoplasmic genetic male sterility. The term itself says that the genes, which gene, male sterile gene, is present both in nucleus as well as the cytoplasm, that is mitochondria. So first time in onion, the cytoplasmic genetic male sterility is discovered by Jones and Davis. So let us understand what do you mean by cytoplasmic genetic male sterility. In this cytoplasmic genetic male sterility, we are having three-line system. We are having three-line system. So if we'll see three-line system means what? One is A line, which is called as the male sterile line. Which line? Male sterile line. Another is male sterile line means this is both the cytoplasm as well as nucleus. Both contains male sterile. Okay. Another is B line. B line is also male fertile. B line is the male fertile, but it is controlled by cytoplasmic gene. Cytoplasmic gene, that is fertile gene. Here cytoplasmic gene is sterile gene is present, and here the nucleus contains the recessive gene. Recessive gene, that is R. So here also nucleus contains recessive, that is R. So R means if nucleus, R means the nuclear gene. The nuclear gene is represented by capital R and small r. Capital R means it will restore the fertility. Means if R is present, then it is fertile. If a small r is present, it is male sterile. In which part? In the nucleus region. Okay. Another is R line. R line is called as the restorer line. It is called as the restorer line. A line is called as the male sterile line. B line is called as the male fertile line. It is also called as a maintainer line. It is also called as maintainer line. What it maintains? It maintains the male sterility. It maintains the male sterility. R line is the restorer lines where the nuclear gene is both a capital R, that is homozygous, and dominant R is present. That means R line is male fertile. It is male fertile. Whatever may be the cytoplasm may contain sterile gene or may contain fertile gene, but whatever may be the cytoplasm is present, gene is present, but the character is expressed by the nucleus gene. Okay. Remember, whatever if cytoplasm contains fertile, but the nucleus gene contains fertile, then it becomes fertile. If the cytoplasm contains sterile, but nucleus gene contains fertile gene, that means the plant becomes fertile. So let us come to this diagram. So let us first understand two things. One is maintenance, another is hybridization. First, maintenance means what? Maintenance of what? Maintenance of male sterile. There is female. Means we want to produce more and more female lines. Okay. So this is called as A line. If I will cross between A line means what? Male sterile line. A line means male sterile. This line is B line. B line is called as the maintainer line. B line is called as the maintainer line. It is male fertile. So why we are going for cytoplasmic genetic male sterility? Because in case of maintenance, we are following which part? CMS part. Which majorly we are following the CMS part. But while hybridization, we are following the GMS part. CMS means we are mostly focused upon the cytoplasmic gene. So in which case, in case of maintenance, we are crossing between A line and B line. So in case of A line, the nucleus contains smaller. B line also contains a smaller. So that is why A line as well as B line, both are isogenic line. Both are isogenic line. Isogenic line means the nuclear gene, this is nucleus. So the nuclear gene contains the same gene and it contains the recessive allele. So it becomes male sterile with respect to nuclear gene. It is a recessive form. Okay. But with respect to the cytoplasm, the male sterile is governed by, I have told you, it is governed by the cytoplasmic gene, that is AS. So here the cytoplasm mitochondria A line contains which gene? Sterility gene. This B line contains fertility genes. That is why it is male fertile, though smaller is present, so smaller is recessive. This one is recessive. Okay, if a capital R is present, then it becomes fertile, otherwise it is recessive. Okay, so now, so here the fertility level or fertility status is governed by the cytoplasmic gene. A line becomes male sterile because cytoplasm contains sterile gene. B line is male fertile because cytoplasm contains male fertile. So that is why whenever I will cross A line and B line, what I will get? I will get small r from here and another r from here. So the nucleus becomes a recessive gene, smaller. But as I have told you the concept or the principle, the cytoplasm will come from the female part because male sterile means this is your female parent. So mostly the cytoplasm will come from the female parent. So here the S will come. So what will be the offspring? The offspring will be male sterile. So here it acts as a female. So whenever I will cross A, B, what I will get? I will get more amount of, this is which line? I will get A line. I will get A line. Means what is the maintenance? I want to produce more and more female seeds, more and more A lines by crossing A and B line. That is why B is called as maintainer line. Maintainer means it will maintain the female lines or A lines. Okay. Then next come to hybridization. So in case of hybridization means what? Hybridization means we are going for two parent. One is female with that is A line which is completely female. A line, or we are saying male sterile. So male sterile means I have told you this one A line. So recessive smaller nucleus gene and the cytoplasm contains the sterile gene. But hybridization means I will cross with R line. Which line? R line. R line is called as the male fertile. R line is called as the male fertile. And this male fertility in case of R line is governed by which gene? It is governed by nuclear gene. Remember the difference. In case of R line, that is capital R, where both R is present, dominant allele, so it becomes male. So it acts as a female, whereas R line acts as a male. And if your cytoplasm contains capital R, or any capital R smaller, or it may be, suppose here it is capital R smaller, then also it is becomes male fertile because capital R is present. Means R is dominant. Okay, restore the fertility. So whatever may be the cytoplasm, whether it is sterile cytoplasm or fertility gene is present in the mitochondria, no problem. If it is sterile, then still the R line acts as a male fertile because the nucleus gene will dominate over the cytoplasmic gene because capital R is present. And if cytoplasm is fertile, then no problem, all are fertile. So if I will cross A line and B line, A line and R line, so A line it is a female, so what will happen? The cytoplasm will come from the female parent. So here cytoplasm is S. But here one allele come from female parent, one allele come from the male parent. So here it is, though cytoplasm is the sterile gene is present, but your nucleus contains the heterozygous condition and it is capital R. Hence, it acts as a male fertile. It acts as a male fertile. Why? Because capital R dominant gene is present. And what is the, so it is male fertile. So what is the role of restorer gene? It will restore the, you can see here the cytoplasm remains same with respect to the female parent, but still the offspring, which is called as the F1 hybrid. This F1 hybrid is male fertile. Hence R line is called as the restorer line, because what is the function? R line will restore the male fertility. You can see the female parent, female is male sterile, but the offspring is male fertile. So this male fertile offspring is restored by using the R line. Okay. So in your breeding program, what we are going for? We are going for suppose in your breeding program, in case of rice field, suppose we are going for rice field. Suppose I will raise one R line, then 6 number of which line? A line. See, 1 R line and 6 number A line. Again one rows of R line. Again 6 rows of which line? A line. So 1:6. So either 1:6 or 1:8. So what will happen? So whenever I will, so this you can see A line is what line? Male sterile line. R line is male fertile line. This is R line is male fertile. So here the pollen will fall over the what? The male sterile lines. And now I will collect the seeds. Formed from the A line, and this A line contains which seed? This one, that is F1 hybrid seed. The offspring, it contains the F1 hybrid seed. Okay. So hybridization program, we are using 3-line system: A, B, R line. B line is called as maintainer line to maintain the sterility. If you want to produce more amount of A line seeds or female line seeds, because you can see here what we need. We need more amount of A lines, A rows. So more amount of A lines will be produced by crossing these things. And to produce F1 hybrid, the seeds will be collected from the A lines. You can see the seeds from the R line should not be F1 hybrid. Okay, but the seeds collected from the, because R, if the R which is male fertile, so in this case cell pollination will be there. Here you can see, in case of rice, what is happening? So it is your, this is your anther. In case of R line, the pollen can fertilize the ovule, because it is which pollen? It is functional pollen. So it can produce, it can fertilize the ovule, and it is called as the cell pollination. R line. But in case of A line, anther is produce the pollens, but this pollen is non-functional. So this pollen cannot fertilize the ovule. Okay, hence which pollen will fertilize the ovule of the A line? The pollen which come from the R line, because R line pollens are functional. So these pollens will fall over the A line, because the A line itself pollens are non-functional. So it is male sterile. Okay, so just collect the seeds from the A line, which are called as F1 hybrid. But do not collect the seeds from the R1, because R1 seeds itself, it is a seed originated from the cell pollination. Okay, so now just collect the F1 hybrid seeds from the which portion? From the A plants, not from the R plants, not from the R lines. Okay, and these hybrid seeds, what are the seeds I will collect from here? The seeds called as F1 seeds, hybrid seeds. The seeds should be used only once. Why? Because it contains capital R small r. So if I will use this, suppose I am going for a hybrid seed production and I will use this seed. Now I will sow the F1 hybrid seeds. So now what will happen? Now they will go for S because these are male fertile. The F1 hybrid seed go for self pollination and they will produce what? Capital R, capital R, smaller, smaller, smaller. Both homozygous as well as heterozygous offsprings will be produced. So here you can see 50% will be heterozygous and rest 50% will be homozygous. So it is a mixture of homozygous and heterozygous. What is the offspring of F1 hybrid? The offspring of F1 hybrid after selfing. Hence it is called as inbreeding depression. So that is why whenever we are using the F1 hybrid seeds, you should use only once. And if you will use this F1 hybrid seeds, again you will use the seeds which will come from the hybrid plants. Then again and again you are going for using the same seeds. Then they will go for selfing, which will go for the inbreeding depression. So you should collect the F1 hybrid seeds from the A lines, and these F1 hybrid seeds should be distributed to the farmer. This is commercially used F1 hybrid seeds. To the farmer you should distribute these seeds, and you should tell the farmer that they should use this hybrid seed only once. They should not keep the offsprings of the hybrids. Okay. So CGMS line is mostly used for seed production as well as the vegetative, where the plants where economic part is vegetative part. But CMS, we have told you, the CMS line is used only for the crops having economic part is vegetative. And one important thing for CMS line is the CMS where only gene is present in the cytoplasm. So it is not environmental sensitive. It is not environmental sensitive. Like GMS, it is environmental sensitive. CGMS is also somewhat environmental sensitive, but CMS is not environmental sensitive. Okay. So in commercial production, you are using this hybrid seed production. Okay. So thank you so much. So if any doubt, then please write in the comment section. Thank you.